fungal disease which attacks the rust crop. And so cereals such as oat, but also wheat and barley and so on, they're very, very important worldwide, and they make up to more than 50% of global calorie intake. So with cereals being such an important part of the global food supply, diseases such as stem rust will have a very large economic impact worldwide. So just to give a numerical value on that, wheat stem rust, which is very similar to oat stem rust, but it infects the wheat crop, can cause up to $200 million worth of damage in a year of a particularly bad epidemic, and that's just in Australia alone. So as you can imagine, with the kind of economic impact that diseases like stem rust cause, it becomes a very important question for us to try and understand exactly how these diseases work, and in particular, biologically, how they interact with the host plant. So our project was kind of split into two parts. The first part was kind of trying to piece together the genetic information of the oat stem rust fungus. And so we started with raw data from the BGI Institute in China. And they sequenced the oat stem rust fungus, and this produces these sort of short strands of DNA at the top there, which then reads. So these are just sequences, AT, CG, I'm sure you've all seen before. So our focus in this part of the project was
I did warn him not to use too many. I said, we're going to be really dumb, we're not going to use talking to people about all these terms. I said, don't use any of those, we're not going to understand.
So our task in this project was to take these short sequences of DNA and to try and join them together as a sort of a jigsaw puzzle, in a way. So this is a process that biologists call genome assembly. And so in our particular project, our raw data consisted of about 78 million base pairs. I mean, that's just a big number that maybe has not a lot of significance. But I mean, you can imagine that it's kind of a really computationally expensive task to try and assemble this jigsaw puzzle. So the process of genome assembly sort of relies on a bunch of really complicated prediction algorithms and whatever, and they would have taken days and days to run on a normal desktop computer. But when we ran them through Artemis, they took at most a couple of hours, so that was really useful. Can we go back a slide?
Sorry, mate.
Right. So once we had our assembled genome, the second part of our project was to try and find the location of genes within the plant, within the genome. So a gene is basically a little sequence of DNA which codes for a specific protein, and these proteins are what do the actual work inside the plant, inside the organism. So in an ideal situation, we'd be able to have access to the actual oat stem rust fungus itself, and be able to sequence the proteins directly and then map those back onto the DNA sequences. But in our particular project, we didn't have that resource, and so once again, we had to rely on prediction algorithms which worked just on the DNA itself. And once again, these algorithms would have taken a long, long time to run on a normal desktop computer, but through Artemis, they were much, much faster. Yeah. So this particular area of research is in very early stages at the moment. We're still at the point of trying to understand exactly how this disease works. In the future, the sort of ultimate goal of research like this is to try and understand the disease well enough so that we can create genetically modified oat crops which are resistant to these sorts of diseases. Now, I just want to say a few words about how useful Artemis really was in our project, given how big of a part it was. So obviously it's a very, very powerful system, and it allows us to run complicated algorithms in much less time than what they would have used to take. And also it's actually a really easy to use system. So as a user, what you see is pretty much a Linux shell. And that's something that I'm sure is familiar to a lot of people, and so it becomes a really easy system to use. Perhaps in the future, maybe a possible improvement would be to build a better sort of UI, or to build a better way to interact with it, because I mean a lot of researchers in different areas don't have the kind of Linux experience that you need right now. So overall, Artemis is a really nice system. It's really useful for our project, and I hope that with the expansions and whatever coming along in the near future, it can still be a really useful resource for researchers in all areas.
Takes a lot of courage to
Good work, David. Good work.